Welcome back. Today we're diving deep into the incredible anatomy of hearing and equilibrium. Let's get started.
The human ear is divided into three primary sections: the external, middle, and internal ear. Each part serves a unique and vital function, working together to capture sound waves, amplify vibrations, and translate them into signals for the brain while maintaining our sense of balance. Let's identify the structures and functions of the ear, focusing on our models and histology slides. Part A, the external ear. Starting with the external ear. First, the auricle, or pinna, which collects sound waves. Next, the external acoustic meatus, containing ceruminous glands to protect the canal. And the tympanic membrane, which vibrates in response to sound. Part B, the middle ear. Moving into the middle ear, we have the tympanic cavity, the three ear ossicles, the malleus, incus, and stapes, which amplify vibrations, and the pharyngotympanic tube, which equalizes pressure. Part C, the internal ear. In the internal ear, we find the osseous labyrinth, filled with perilymph, and the membranous labyrinth, filled with endolymph. Part D, subdivisions and equilibrium. There are three main subdivisions: the cochlea, housing receptors for hearing, the vestibule for static equilibrium, and the semicircular canals for dynamic equilibrium. Part E, deep dive into the cochlea. Looking closer at the cochlea, the scala vestibuli and scala tympani both contain perilymph. The cochlear duct contains endolymph. Note the tectorial, basilar, and vestibular membranes. These support the spiral organ, featuring inner and outer hair cells that trigger the cochlear nerve. Finally, observe the oval and round windows. Part F, the vestibular apparatus. For balance, the vestibule uses the utricle and saccule. Here, otoliths within a gelatinous material move hair cells to sense gravity. In the semicircular canals, the ampulla contains the crista and cupula, which detect rotational acceleration.
Now let's look at how we maintain equilibria. To stay balanced while standing, the brain requires input from at least two of the following three senses. First, proprioception, which is the internal sense of where our body parts are in space. Second, vestibular function, which uses the inner ear structures we've identified to monitor head position. And third, vision, which allows the eyes to monitor and help adjust the body's position. In the lab, we perform the equilibrium test, often called the Romberg test, to see how well the body maintains balance when one of these senses, like vision, is removed. Because these pathways are so sensitive, balance tests are also a primary clinical tool used to detect alcohol and drug-induced driving impairment, as these substances disrupt the brain's ability to process these vital sensory signals.
Next, we examine nystagmus, which is the involuntary rapid movement of the eyes. We test this using the Barani test. By rotating a subject in a chair and then stopping them abruptly, we can observe their eyes oscillating. This happens because the fluid in the inner ear, the endolymph, continues to flow even after the body has stopped moving. This test confirms that our visual system and vestibular system are intimately linked to help us maintain a steady gaze during movement.
Next, we conduct laboratory tests to differentiate between conduction and sensory neural deafness. Conduction deafness occurs from a physical blockage in the outer or middle ear, while sensory neural deafness stems from damage to the cochlea or auditory nerve. We evaluate this using two tuning fork tests. The Weber test checks for lateralization by placing the fork on the midline of the skull, while the Rinne test compares bone conduction on the mastoid process directly to air conduction outside the ear canal to locate the source of hearing loss.
Thank you so much for watching. I hope you learned something new today about the fascinating anatomy of hearing and equilibrium. Stay tuned, and we'll see you in the next video.